Dear students, in today's lecture we are going to talk about how the genes of a bacteria or any other microbe for that matter evolve over time as through replication and through succession in environment. So, one of the major source of changes, genetic changes for microbes are mutation, and the other are the drifts, that genetic drifts that happen within microbial communities. So, today what we are going to do is we are going to go through the reasons why genes evolve over time, and remember, genes are very important because genes are the reason, or let us say genes are the information code of any microbe, and they decide what proteins can be expressed, what activities a microbe can do or not do, and thus it is very important for us to characterize the genes and know. When genes change through succession or through genetic drift, then the characteristics of the microbe also changes, and today we are going to explore the genetic evolution of microbes. Alrighty. So, previously I have told you about the basic paradigm of microbiology, where the DNA stores the information and then the RNA is transcribed from DNA. So, remember, if DNA is, let us say, 5 megabase pair big, RNA does not have to be 5 megabase pair big. In fact, it could be only, let us say, 600 base pairs, and thus in this case RNA would be a transcribed copy of one gene. In bacteria and prokaryotes, many a times a messenger RNA can have multiple genes, can be transcription copy of multiple genes. The difference between DNA and RNA for revision is that here in DNA we have ATGC, here we have AUGC. So, instead of thymine nucleotide we have uracil nucleotide. Another thing you need to remember is that RNA are usually short lived, and as their name suggests that we are talking about mRNA, messenger RNA, their main job is to serve as a template from which the protein can be translated. Now, when the cell carries out this process of transcription and translation, there are various steps through which the cell can control how much protein is produced and when. Now, this is very, very important to understand that if a DNA is present, let us say DNA is present for methanogenesis, it is not necessary that the proteins required for methanogenesis would be expressed. It is quite possible that there is some control in transcription process, and the gene that codes for methanogenesis or the proteins required for methanogenesis will never be transcribed to RNA. Now, this is very important because this forms as one of the first control for a cell, transcription control. Using different enzymes or other signals, the cell can force a part of DNA not to be transcribed. The second step of control for the cell would be degradation of RNA. As I mentioned, RNAs in general are short lived compared to DNA. Now, the interesting thing here is that different kinds of RNA have different lifespans. Some messenger RNA are short lived and some of them are long lived. Now, this is another step of control for microbes, because the short lived mRNA will have less time to express protein. So, if there is a particular protein that is required in less quantity, all the cell needs to do is ensure that the messenger RNA, which is a transcribed copy for the which will act as a template for that short lived for the protein required in short amount, this mRNA should have shorter life. The protein that is required in larger quantity, obviously its mRNA should have much longer life. So, this is the second step of regulation. The third step of regulation of protein formation is in the translation step. Even if I have made mRNA, let us say transcription is successful, the mRNA has not destroyed yet, even in the translation step, when a copy is made from mRNA, there is quite possibility for the cell to control how much protein is expressed. For example, let us say initially there was a need for a particular protein, let us say hist C protein, which helps in degrading histamine. Now, the hist C protein, by the moment the hist C protein is enough, the cell can stop further translation by controlling this translation step. Again, here it can use different enzymes, signals to inactivate the mRNA so that further protein cannot be made. There is a fourth step of control where, let us say, initially there was lot of lactose and the cell expressed proteins to digest the lactose, to break it down and use it as energy source and carbon source, but all of a sudden the media has changed and now there is no lactose, the lactose has disappeared. So, these lactose proteins that help degrade lactose have to be inactivated or rendered useless. In this case, the cell, there is no point for the cell to stop transcription or to destroy the mRNA or to stop a translation, because the protein has already been expressed. In this case, what the microbe can do is render the protein ineffective. Now, there are many steps of doing this. There is, in this course we will not be going through the regulatory details, but this is a very important information to understand the limitations of studies that are based only on DNA or only on RNA or only on proteins. In fact, as I mentioned in the last lecture, we need to account for all of them, DNA, RNA and protein, to get a realistic picture. So, to summarize, presence of protein gives me an idea of activity in recent past or in present. Presence of RNA, mRNA gives me an idea of activity in now, because mRNA are usually short lived and protein is longer lived than mRNA. So, mRNA is a snapshot in time. Protein also captures some of the past, because most proteins live substantially long. DNA lives the longest. DNA gives me the potential, which may or may not be translated into an active protein. Alrighty. So, now let us come to cells. Now, as microbes evolve and as they succeed from parents to daughters and again daughters and so on and so forth, they undergo changes. They can be because of natural reasons, the natural error that happens when cells replicate. So, when the DNA replicate and the parent cell has grown twice the size to now split into two daughter cells, it is quite possible that there is an error and in making the copy of the DNA, and thus some mutation has been introduced. Now, most of the mutations that are introduced in such way are not meaningful, they do not really harm the cell. So, these not harmful mutations over time can collect, and eventually we might have speciation, where the daughter cells are very much different from the original ancestor. So, we are going to look into the mutations first, understand how genetic evolution occurs, how even anthropogenic xenobiotics such as heavy metals and at times radiation can actually increase the rate at which mutations happen, how we study them, and how and why mutations are so important for understanding the microbiology. So, let us define the terms. Wild type. Wild type is a microbe which implies that it has been isolated from the environment and it has not been mutated. So, it is the original or the wild type strain. When one of the gene or if there is any genotypical difference in the wild type's daughters, then they are known as mutant. So, let us say I have wild type strain 100, then the mutant has some differences and then it could be strain 102 or 103 or something like that. So, that is wild type versus mutant. Then we have screening versus selection. So, screening versus selection, it is quite possible that even when the mutant is genotypically or in terms of genes different from the parent cell or the wild type, it is quite possible that it has similar behavior, because this mutation does not cause or does not render any important protein useless or does not cause damage to the cell. In this case, both the mutant and the wild type will behave very similarly. So, we cannot screen them, but over time we can grow enough colonies and we can notice that okay, even though they digest the same food, even though they are resistant or susceptible to same antibiotics, but there are slight differences in the shape of the colonies they make. So, they do everything as like each other, the wild type and mutant, but there are certain surface differences, morphological differences. So, if you grow enough colonies, you can pick out the mutant from the wild type. This kind of process is called as selection. At times when a mutant is made, the difference in the qualities are so clear and clear cut that we can screen out the mutants from the wild type. One example I want to give is cloning process. So, the routine cloning that we do in our laboratory for amplifying a particular gene and sequencing it, or for expressing a particular protein that I am interested in the routine cloning process, usually the genes that the competent cells that successfully pick up the gene that I am interested in cloning, they become resistant to an antibiotic, typically ampicillin or kanamycin. So, if I take the I will show you by diagram. So, let us say this is my box of bacterial cell, some E. coli, harmless E. coli. Now, all of these E. coli have certain genes in them, their chromosome, all supercoiled chromosome. Now, all of them are competent. So, when I talk about competent cells, we will talk about it in more detail either in this lecture or in next lecture, but all of them are competent cells, which means that they are ready to pick extra DNA lying around them. So, if there is any DNA fragment lying here, they can pick it in and bring it inside their cell. These cells are competent. So, let us say there are competent cells lying here. Now, in the typical routine cloning process, we introduce the genes, the genetic element we want to clone, we put it around competent cells, we create the right chemistry, the right temperature, the right conditions for these competent cells to pick the DNA fragment up. And when they have picked up the DNA fragment, it so happens, I mean the way these E. coli have been designed is that they become resistant to an antibiotic. Let us say now after they have picked up, these E. coli are resistant to kanamycin. Now, it will so happen in my experiment that not all E. coli will pick up the DNA I wanted them to pick up. So, some of them have not picked the DNA I wanted them to pick up. So, they are just sitting here. Now, if I have a plate and in this plate I have added kanamycin, I have added kanamycin at the right concentration. So, only the cells that are resistant to kanamycin will grow in this plate. So, if I scoop this out and I pour them here, only those cells will be able to grow who have picked up the right genetic fragment, and the ones that have not, they will die out. So, the ones that have not picked the genetic fragment, they will die out, and the ones that have picked will successfully grow on the plate. Such a process of removing, now remember, because the ones that have picked up the gene, their genetic composition has changed, some new addition has been made. So, they are mutant, whereas the earlier one was in a way wild type, even though in case of routine cloning it is not wild type because it is not picked from environment, but this is the original one. So, when we isolate the mutant from the wild type on basis of their qualities such as resistant to an antibiotics, or being able to degrade some sugar, or being able to synthesize some protein or not synthesize some protein, this process is called as screening. So, screening, when we do the main difference between screening and selection is, in selection both the wild type and mutant can grow, but in screening only the mutant can grow or only the wild type can grow. So, one of them is removed and thus it is easy to isolate them. Alrighty, the next term we need to understand is nutritional auxotroph. Sometimes when a mutation happens, let us say there is a gene HIST-C1, HIST-C2, histamine according genes. So, these genes, if they have been knocked out, then the cell can no longer synthesize histamine, and thus it requires histamine to grow successfully. Such mutants are known as nutritional auxotroph. So, nutritional auxotrophs are those microbes that are mutants from their wild type and they require a supplement to survive, and the initial wild type is called as prototroph, because prototroph is what we are comparing the nutritional auxotrophin. Now that we are talking about mutation, one of the words that I have been throwing around a lot is genotype, genotypical differences. So, as the name suggests, genotype is referring to genes. So, any genetic differences, we are talking about genotypical differences. Phenotype is your morphology and observable attributes, things that we can observe such as morphology, motility, color. Now, both of them are very helpful when we do screening or selection of microbes, but let us try to understand mutation in terms of genotype and phenotype. So, if the daughter cells differ genetically from the parent cells, then this is genotypical difference. Now, a genotypical difference, as we have mentioned, may or may not have an observable attribute difference. So, we might have knocked out few genes, but they are not essential genes, and we really cannot observe the difference between wild type and mutant. It is quite possible that it is only single nucleotide difference, and it does not make much difference in the behavior and the observable attributes of the cell. So, we might have a genotypical difference, but without any phenotypical difference. However, in general it is nearly impossible to have phenotypical difference without any genotypical background. Microbes usually they behave as their genes ask them to. So, if there is a phenotypical difference, for example, there are two Pseudomonas aeruginosa, one makes really good biofilms, the other does not. So, we might know that the mutant which does not make biofilm, so that is a phenotypical difference that perhaps has genotypical difference compared to the wild type that makes beautiful biofilms. Now, one thing I want to show you, the genotypical differences are often written on name by the name nomenclature is often done like this. Usually you will have a 3 alphabet written in italics. Here it is HIS followed by a capital letter and maybe a number or not. So, his C1 is the first mutant in his C gene. So, this is one of the way of writing genes. Now, let me tell you. So, when we are writing in computer or in words or any electronic media, the way we write gene names is we italicize the gene name. Now, C is not italicized, only the first 3 letters are, and if you are writing it by hand, then you need to underline it. Okay. So, if I were to write his C, which is a gene by the way, by hand in a handwritten document, I would write it like this, underlining the name. Now, if there is a change, a genotypical change in this particular gene, I make this is wild type, let us say, and there is a genotypical difference, then I can call the mutant as his C1, and let us say I get another mutant, I can call it his C2, and so on and so forth. Now, if we have phenotypical differences, then we do not use italics, we write them normally, and we often write plus or minus to show phenotypical difference. Remember, phenotypical differences are observable difference. So, ability to synthesize histamine could be HIS plus, yes it can do it, or HIS minus, no it cannot. So, this is your wild type and this is your mutant. Now, dear students, take a careful look at this table. Here I have listed 5 different genotypes, 5 different 4 mutants and the wild type gene. So, the wild type gene is his C and these are the 4 mutants, and this is my convention of writing it by hand. Now, it is quite, now the wild type is his plus. So, it has the ability to synthesize histidine. There we go, perfect, metabolize histidine, very nice. Now, after I have introduced one mutation, it is possible that it can still metabolize histidine. So, let us say even despite a mutation in his C gene, it can still metabolize histidine, and then when I do another mutation, his C2, now it can no longer metabolize histidine, and thus it is his minus. Clear? Now let us say another mutation, his C3, also cannot metabolize histidine here. Now it is quite possible that I do mutation to his C3 and I create a mutant his C4, and this reverses the effect of the mutation that happened here, and his C4 can now metabolize histidine again. So I have his positive. So note here, some mutants can metabolize histidine, some mutants cannot, but all of them are genotypically different from the wild type. So the genotypical differences may or may not translate into phenotypical difference. Another thing I want you to notice, that once a mutant has lost an ability to metabolize something or to do some action, it is quite possible to mutate it further so as to reverse the effect, so that it starts behaving phenotypically similar to the wild type, even though genotypically it is still different. So even this is a possibility. For example, initially let us say we have a susceptible pathogen and it undergoes mutations, and now it is antibiotic resistant pathogen. It is quite possible that it will undergo mutations and then again become susceptible to antibiotics while still being genotypically very different from the original wild type. Alrighty. So now let us look at mutation. There are two types of mutation you want to pay attention to. One is the induced mutation, the other is spontaneous mutation. So spontaneous mutation, as name suggests, they happen spontaneously without any anthropogenic or environmental influence. So as I mentioned before, during replication, when DNA is replicating, it is possible that there is some error, or let us say there was an error and now the microbe is trying to repair the genes. Yes, my dear students, note this very carefully, that microbes have the ability to repair genes. So when microbes are trying to repair the genes, it is quite possible that in the repair mechanism they introduce some errors. Alrighty, and then in induced mutations we have here, in induced mutation we actually cause mutation by changing the chemistry around them or by exposing them to mutants, by exposing them to chemicals or radiation, environmental factors that encourage mutation. Now we can broadly classify mutations into three different forms: silent mutation, missense mutation and nonsense mutation. So when a mutation happens but it does not change phenotypical characteristics, the cell can do the same thing it was doing earlier, there is no phenotypical difference or activity based difference between the wild type and mutant, then these mutations are called silent mutations. Thankfully, most of the mutations on earth in our life are silent mutation. Then we have missense mutation. So in missense mutation, what happens is that we get the protein, but the protein is faulty protein. So protein can still somewhat do the job, but it is not as good as the wild type, and this is missense mutation. Now in nonsense mutation, what happens in the mRNA, messenger RNA, because of mutation a stop codon is introduced. So a stop codon would be UAG. So in mRNA where something else should have been, for example, let us say we have a long mRNA chain, this is your messenger RNA, and here you were supposed to have UAA, alright, but we have a mutation and now instead of UAA you have UAG, correct. So when your ribosome, starting at the beginning of the mRNA, it is making protein as it moves. So this is a protein chain which it is making. By the time it reaches here and it has made some level of protein, it will read this as UAG, and UAG happens to be a stop codon. So ribosome says, okay, the gene stops here, I should fall off. So it drops here and we are left with this size protein, and all the rest of the mRNA that was supposed to be translated is not translated, and thus the protein fragment that we have is incomplete and it cannot do its work. In this case we call such mutation as nonsense mutation, because the ribosome drops prematurely because it has read a stop codon. So whenever a mutation results in ribosome reading a stop codon, it is a nonsense mutation. These are very dangerous. Okay. So let's look at this picture here. This is your original DNA. It underwent mutation. So originally it was TAC from 5 prime to 3 prime end and ATG from 3 prime to 5 prime, perfect complement to each other. They all underwent mutations of different types. So when TAC mutated into AAC and ATG became TTG, it coded for this particular codon coded for asparagine, and thus you have a faulty protein. This is missense mutation, right? Because actually the original was supposed to code for tyrosine, but now it is coding for asparagine. So this is missense. The faulty protein might do a good job, might do a decent job, might be fairly good or might not be effective in doing the job that is required to do, but because it is just one amino acid in a long chain of proteins, it is quite likely that it will be a faulty protein but not deadly. Now if TAC undergoes mutation in the third nucleotide and TAC becomes TAG, we get ATC, and this TAG is read as UAG. Now UAG, as I mentioned, is a stop codon, so the ribosome drops off and the protein is incomplete. This is nonsense mutation and it is very dangerous for the cell. On the third level, the TAC becomes TAT, it becomes ATA. Now TAT is a codon, this is the mRNA UAU, and it so happens that UAU also codes for tyrosine, which the original UAC was doing, and thus there is no difference, that it will still make the normal protein that the wild type would make, even though there is a genotypical difference. Such mutation is called as silent mutation. Now if you look at the left panel here, we have a very beautiful image of how we select different microbes that have mutated. So here we have auxotrophic mutant, so nutritional auxotrophy. So we have a velvet surface and this is your medium that contains histidine, and the ability to metabolize histidine is in the hist-C protein. Now the both medium, one of the medium has histidine, the other does not. So the microbes that need histidine to grow will grow only on this one and not this one. So auxotrophic mutants will only grow on this plate and not on this plate, whereas the wild type will grow on both. So what I can do is, this is my medium that is containing histidine, it has both auxotrophic mutant and the wild type. I can use sterile velvet surface to pick these colonies together and then I can plate them on both. So I can just do like a stamp, I can stamp them on both. Now the medium that contains histidine, everything will grow, but those auxotrophic mutants that require histidine to grow will be missing here. So this way I can pinpoint, oh, this is a missing spot, so this must be my auxotrophic mutant. So this way we can screen out our mutants. Alrighty. Now let's look at different kinds of mutations. We have so far talked about missense, nonsense and silent. Note that these are all single nucleotide mutations. So they are all point mutations happening at particular point. Very dangerous mutations that happen are frame shift mutations. So in frame shift, now remember that DNA is converted into mRNA and then the mRNA is read by ribosome in group of 3. So they are called codons. In frame shift mutation, let us say it starts with UCA, but now it will skip one step forward or one step backward, and when this happens, then the problem begins, a real serious problem, and the entire protein would be faulty. These are very dangerous mutations. So for example, let us say we had UCA, UCC, UAC, GGCU and so on and so forth. We should have had different amino acids as I mentioned here, but now what happened is this U has been removed. So okay, if we let's start from here. This is my original wild type. So we have UCA, CCU, AUG, GCU. So the ribosome binded here at ribosomal binding site, and now it is reading in 3, and it will read UCA, CCU, AUG, GCU and add the amino acids as required. Now let us say we add a uracil here. So this will create a frame shift mutation. How? So when we add a uracil here, it is still reading the first 3 as UCA, but now instead of reading CCU, it will read UCC, which will code for a different amino acid, and then all of them will shift and we will have very different sequence than what was supposed to be. So this is a frame shift mutation. The frame of reading has shifted. Now on the other hand, we can delete the C. So this C highlighted in red can be deleted, and when it is deleted, Now the ribosome will still read UCA, but now instead of reading CCU, it will read CUA, UGG, CU and so forth. Again a very different protein will be made. Now these frameshift mutations are very deadly because by deleting or adding a single nucleotide, we can have very different protein, almost always a very different protein which does not do the job, or at times it might be even dangerous for microbes. Now this is when we are adding or deleting one nucleotide. What if we are adding or deleting two nucleotides? If we are adding or deleting two nucleotides, we still run into the same problem. However, if we add or delete 3 nucleotides, let us say the ribosome jumps a codon, then one amino acid might be extra or one amino acid might be missing depending on whether 3 were added or 3 were deleted, 3 nucleotides were added or deleted, but it is quite possible that the overall protein, despite being faulty, will do the basic job and thus it will not be deadly. But frameshift mutation, if it is an essential protein, it will be deadly. So now let us look at some of the mutants, some mutagens and how they act. So we have base analogues. So base analogues are very similar to the base, nucleotide base. So for example, we have bromuracil, it acts like T, like U, but it has a bromine which normal uracil does not, and it incorporates like T and it causes faulty pairing with G, when it should have actually paired with A, but now it will pair with G. As a result, we have a very different transcription. Aminopurine, it acts like A and it faultily pairs with C. Then we have chemicals that react with DNA. For example, nitrous acid HNO2, it will deaminate A and C. Hydroluxamine NH2OOH, it reacts with cytosine. Then we have alkylating agents such as ethyl methane sulfolate, they put methyl on G and they causes faulty pairing with T. And then we have bifunctionals such as mitomycin, nitrogen mustard, nitrosogunadine. They cross link the DNA strands and they completely destroy your DNA structure, and the faulty region is then excised by the DNase, which causes a lot of genetic damage. Then we have intercalating dyes. The intercalating dyes, they have a very interesting way of destroying DNA or causing mutation in DNA. What they do is, so what these intercalating agents do is, let us say you have DNA. So this is a schematic of double helical DNA, and forgive my poor drawing, but DNA looks like a very beautiful oppositely paired double helix, one helix running down to up and other running up to down. But let us say you have this double stranded double helically packed DNA. Now intercalating agents will come and intercalate here. So what they do is, so now you have an intercalating agent here, and thus your DNA becomes, there is a gap in DNA. So when you start replicating it or when you start transcribing it, then often one nucleotide or some nucleotides are deleted. So this causes mutation too. Now ethidium bromide and other dyes that are used for looking at such as cybergreen, dyes that are used for looking at DNA, all are intercalating agents, and when transcription happens, when translation happens, they all result in mutation. Now let us look at radiation. Ultraviolet rays are nasty and they are used for disinfection because they cause pyrimidine dimers. So pyrimidine dimers means they will form thymine dimers often, sometimes they will also form cytosine dimers, and when ribosome interacts with thymine dimer, so uracil dimer or cytosine dimer, then it drops and the replication process, and so sorry, not ribosome, DNA tag polymerase, when it encounters thymine dimers or cytosine dimers, it drops and replication stops, and thus anything forward from the pyrimidine dimer will be deleted. Then we have ionizing radiation. So ultraviolet is non-ionizing radiation. Ionizing radiation, it is a free radical, they make free radicals because they ionize, which attack DNA, they break the chain, they destroy the structure. Now the cell is not defenseless when it interacts with these mutagens. it has DNA repairing system as I mentioned earlier, and usually it is controlled by these two genes Rec A and Lex A. So, these are the proteins Rec A and Lex A, and what this little schematic is showing you here is the SOS response to replication damage. So, all of them will call replication damage, all the mutagens. So, what will the cell do when it notices that there is a replication damage? So, when replication damage is noticed, it stops the replication process and it starts looking at DNA damage. It activates the protein Rec A, which deactivates Lex A. So, Lex A is a stopper on SOS response, and it is important to have Lex A here, because if you do not have SOS response, SOS has the possibility to save the cell from the DNA damage, but also create DNA damage. So, SOS introduces lots of error in the genome. So, the cell has Lex A to stall it, to stop it from happening, but when Rec A is activated, Lex A disintegrates, it cleaves, and it is no longer stopping SOS response, and the SOS genes they get activated and the DNA damage repair starts. So, dear students, we have already talked about these three different mechanisms of bacterial gene transfer. So, apart from mutation, apart from selection, another way in which microbes change their genetic qualities are by horizontal gene transfer. So, we have talked about transformation just now, where the bacteria they pick up the DNA fragments from the neighborhood. So, this is the blue fragment are lying from a dead, have been released from a dead cell, and the recipient cell will pick them up. This is transformation. In conjugation, two bacterial cell, one which is the fertile will make a bridge, and from the bridge the DNA will be shared. So, both of them will have a copy now. It is a very beautiful process. We will talk about it a little bit more in detail in the next lecture. In transduction, virus are used. So, the virus here, what they do is they pick up the genetic fragment, some bacteriophage, and when they go to the recipient cell, they give away that genetic fragment to it. And we will talk about transduction in detail also, but this is just a basic revision. This is another way through which bacteria change their genetic sequence. The next way in which bacteria undergo genetic differences is recombination. So, this is a very beautiful diagram of recombination, and I have some information here that you might read if you are interested. Basically, what we have is that endonuclease will create a nick, it will create a nick in the DNA, and once this has been created, it is quite possible it will overlap with the another DNA using Rec A protein. So, if you remember from here, once Rec A has been activated, SOS system is on. So, SOS system is on in this microbe, and it will create gap in the another double stranded DNA, where this nick part can come here and they can exchange. So, we can have different kinds of patches and splices. This is a recombination. So, this is all for today, and just to revise, in today's lecture we talked about how the genetic evolution takes place. Basically, we talked about two directional movement in genetic evolution. One is vertical, when going from parent to daughter we have succession, we have mutations, and they cause different kinds of damage or do not cause damage to the cell. That is one way of changing genes. The other way is horizontal movement. So, in horizontal movement we have a transformation, transduction and conjugation, and the third way is when within the cell we have recombination. So, in recombination some genes might be disrupted either by deletion or by addition, or the operons might be shifted. So, the behavior of the proteins would change, the behavior of the cell would change. So, my dear students, this is all for today. In next class we will take this further and note how when we know how genes are changing, we can understand how close the microbes are genetically, how we can draw trees from it, understand the similarity between bacteria, type the bacteria, and we will take it from there. Thank you so much.